And then, last, post-translation modification. And in particular, when we have a peptidase, this peptidase leaks protein and cut it in different parts. If I use the peptidase in solution, then my final result is a mixture of fragments coming from the protein to digest and from the peptidase surface. When the peptidase is immobilized on the surface, the self-digester is not allowed, and then the only result is a mixture of peptides originating from the protein to digest. There is a particular attention on the technology I use to immobilize the peptidase on the surface. Because it is very, very important in order to guarantee the activity of the peptidase.
Using proteomics, and proteomics in vaccine research has been successful in identifying new vaccine candidates, and this is one paper that was published in 2006 reporting the identification of a new vaccine candidate for group A Streptococcus. And in this paper, a new technology was described that is proteomics and mass spectrometry based. So these methods rely on the shading of the surface of live bacteria with proteases. So this means that we start from a bacterial culture, then we use trypsin or other proteases to cut all those proteins or protein domains that are protruding from the cell surface. So in this way,
very small capillary of 1 microliter. And therefore, in this case, it is possible we develop this strategy to inject 30 nanoliter of cytochrome P450s or substrate. And then we reduce the consumption of the substrate or of the enzyme by a factor of 500 as an example. And the analysis is very simple. We have no separation. It is just an incubation into a capillary. It is not a capillary electrophoresis. It is just a capillary which is inactive and where we have the mixture. When the reaction was done, we flush the sample into an acidic solution in order to stop the reaction. And we obtain exactly the same results as obtained in solution with cytochrome P450s or with human liver microsome. We obtain exactly the same results, but we decrease the consumption by a factor of 500 as an example, which is quite cost effective. And finally, one advantage of this system is so simple, it's just a capillary, just applying a small pulse of pressure without separation, without detection in this system. It is very simple. We have developed in our group a multi-capillary system with 12 capillaries, permitting to perform in parallel 12 incubations. As an example, with 12 different cytochrome P450s or with 12 different substrates. And we have developed with
You can see how the tip is following the liquid here. and again for the organic liquid. Yeah. So the tip is always following the liquid, and you can also calculate the volume of the liquid, because when you have a proper Lapoil definition, then you can hit the liquid level, and then you can in that way calculate the volume that is in your bottle, or in the deep well log, for example. This is the monitored air displacement, the map what I already said. In this way, we are always monitoring the pressure during the aspiration and dispensing, and we have in this way always the option to realize that something was going wrong or something is going wrong during the pipetting step. So the aspiration is okay, means